I hope that you all are enjoying your lunch. I know that some of you have just been served, so please keep eating, but we are going to get on with our program that we have for you over this seated lunchtime, and trying to stay on time because I know people have to move to sessions after this. So this lunch is our way of celebrating 25 years of collaboration at ESIP. To me, 25 years is nothing to take lightly. I actually looked it up, and according to the National Center on Charitable Statistics, 1/3, 33% of nonprofit organizations don't make it 10 years. 10 years. And you probably see it. You know, there are initiatives that spin up and they start up and then they kind of fade away. And so 25 years of ESIP is really, really something to celebrate. Erika gave a talk yesterday about some work that a group of us did on the sustainability of earth science information organizations. And I had the privilege to be part of that work. And I got to meet some amazing pioneers in this space and in this community and hear about the work that goes into making organizations that have lasting impact. We talked about factors for success like leadership and decision-making and agility and business models. We talked about the decisions that get made along the way, the governance. We talked about the skills needed in early leaders and in later leaders. And to me, a lot of it came down to people, right? At ESIP, we talk about data a lot, but it is really the people that make the organization. And they say that as individuals, we are shaped throughout our life by the people we encounter, first by our parents, and then our teachers and our classmates. And I would say that at ESIP and at an organization, that is even more so true. We are shaped by the people who have been a part of ESIP along the way. And I just want to take a moment before our wonderful speakers to recognize the people of ESIP. So many of you know that the early leaders of ESIP were some amazing names, including Martha Maiden. Looking through documents, I saw names like Peter Cornelian, who participated in the work I just mentioned. So I'm not sure if there is anyone, but if there was anyone here who was in the room in 1998 when ESIP was formed, would you please stand? Yes, Rama and Bob. Yeah. Oh, wow. Look at this. Tamara. And I would like you all to stay standing. These people, that is commitment. 25 years, and they're still here with us. That is amazing. So stay standing. Don't sit down. I would like to recognize our past board chairs and presidents. In the early years, they seemed to there was a chairman and a president and then a president and then a chairman. So if you are a past or current board chair or president, would you please stand? We have a running question as to who is a longer serving board president. Was it Ken or was it Dave Jones? We're going to have to do some digging on some minutes to figure that out. So I would like all of our past and current board members to stand. Well, a few more people. Looks good. Thank you. And then I would like any elected leaders, including our program committee and our technical committee chairs, past or present, to stand. I would like to recognize any past or current staff, if you can please stand. Now, these are some people that work really hard, and it's a big group of people. So I would like you to stand if you have ever chaired an ESIP cluster. These cluster chairs work really, really hard. I would like to recognize our current and past community fellows. there is nothing that excites me more than seeing a community fellow that then is employed by a partner organization and they're still engaged in the community. We have past community fellows on the board. So I see some community fellows that are still seated. So past and current community fellows. I see you, Jim. Yep. Please stand if you have ever received an ESIP Lab Award or a Funding Friday Award. Please stand if you have ever staffed or volunteered at an ESIP Data Help Desk. Oh, Shelly got excited at that one. She's like, that's me. I would like to recognize anyone who has ever been a meeting session convener, a speaker, or a poster presenter. What's so funny? You get where I'm going with this. Please stand if you have ever attended an ESIP cluster call or webinar. And finally, if you have ever attended an ESIP meeting, please stand. So I want to thank you all. You can now take your seats. This is community, right? And it takes community to have an organization that lasts for 25 years. Community is not something I take lightly, and I'm really grateful to all the leaders who have come before and built this place. And for those of you who are newer to ESIP, I hope you see with all those categories, there are a million places you can engage with ESIP. You can take that first step of just attending a cluster call or having a poster at our next meeting, or figuring out where you fit. One thing we're doing a little bit differently this year, typically at the end of our annual business meeting, which happens on Friday, we do something called caucusing for our committees. So ESIP's administrative committees, finance, governance, partnership, meetings, and equity, diversity, inclusion, and justice. We like to have partner representatives from our type 1, type 2, and type 3, so the different types of organizations, staff those committees. Those are essential to how we do the work that we do. I don't want to leave that to Friday because I've heard there's a couple people getting on airplanes before Friday, and I don't want you to miss out. So we have up by registration, there are big post-its on the wall. You've got the unconference ideas, but there's also committee signups. I want to thank Tracy Pilone for being an amazing leader. She has already signed up for the finance committee. But I would challenge us if we got to Friday morning and our committees were all full and we were like, oh, we don't have to caucus for committee participants. That would be amazing. So think about where you can give to ESIP. Think about what you want to get from ESIP, and let's find ways to get you engaged. Sign up for a committee or join a cluster. We would just love to have your engagement, not just this week, but throughout the year. So with that, it is my pleasure to welcome Megan Carter, ESIP's Community Director, to introduce our speakers for this afternoon.
All right, so I have the pleasure of introducing our Raskin Scholar. The Robert G. Raskin Scholarship has been awarded annually since 2014 to an outstanding graduate student who has an interest in the community evolution of earth science data systems. The award's namesake, although I never met him, was known for being soft-spoken, easygoing, wise, and especially he was known for his dedication to mentoring aspiring data professionals. It is with great honor today that we award this to Alexis Gerritsen as the 2023 Robert G. Raskin Scholar. Alexis is a graduate student at Tufts University and the Jackson Laboratory, as well as a data scientist for anecdata.org. Her work repurposes neglected data to investigate how communities change over time using a combination of genomics and earth science methods. Alexis is already contributing to ESIP, as she has served as an ESIP Community Fellow for 2 years. She has served as a data help desk expert, and she's just starting her term as the newly elected chair of the Data Stewardship Committee. In addition to her numerous accomplishments, Alexis, like Rob, is known for generously giving of her time to mentor junior colleagues and peers. So for all of these reasons and more, I'm very excited to welcome Alexis here today to speak with you all and to get to present this award to her.
Right. Thank you so much. I'm really excited to be here today to talk with you all. As Megan said, I'm Alexis, and I'm excited to share a little bit about how my data work intersects with collaborative science and open science. There we go. I wanted to front load my thank yous because there are just so many of them. I am so grateful to the family of Robert Raskin for funding this award, but also to the ESIP community, both those that were involved in the selection and the broader ESIP community in general, who have all supported my progression in the field, particularly the Data Stewardship Committee members and the chairs over the past couple of years. I'm also super grateful to the Community Fellowship Program. I was telling Megan that the Community Fellowship was the first grant that I ever got, or fellowship that was I ever got during my graduate school, and it really has launched me into a space that I didn't really even know existed when I started out in earth science. And I'm also super grateful to all of my collaborators, funders, students, everyone who has made this work possible. So I've had kind of a meandering path through the life sciences to find my way to earth science. I started out as an undergraduate at George Mason University, doing my first set of research projects in conjunction with the Smithsonian institutions, where I focused largely on using image data or different types of biodiversity data to think about how organisms exist in our planet. I've kind of moved towards more and more mammal systems, things that look a little bit closer to you and I. But what kind of ties together all of these different organisms is that they're sharing the earth with us. And one of the things that's affecting all of us that share the planet is climate change. And I struggled with what to put on a slide titled climate change at this organization meeting, because there's so many things that you could put here. But one of the things that's been really pressing and immediate this week has been the ways that climate change is affecting our ability to get to the conference and experience it. You know, you can hear the rain on the roof during some of the sessions. It's pretty amazing. And this flooding has been pretty astronomical. But this kind of flooding and changes to direct observable meteorological events is not the only ways that anthropogenic change is affecting our environment. And a critical one is how it's affecting the biodiversity, the composition, and the interactions of ecosystems that we live in. I focus particularly on phenology, which is the study of the timing of natural events and life cycle events within different organisms. So you can think of the timing that the leaves in fall turn red, or when migratory birds return from their summer seasonal homes. And phenology, we've discovered over the past few decades, is incredibly sensitive to climate change. And so as we see warming, these life cycle events are cued very closely by the temperature, the day length, and precipitation. But species can respond to varying degrees to the same changes in a cue. And this can lead to pretty substantial disruptions to ecosystem functioning. For example, if you have a migratory bird returning from its summer home, it might come to the north to discover that its food source has not yet emerged from the eggs. Or similarly, you might see insects looking for flowers to pollinate that haven't yet bloomed. And then that flower is left without a pollinator when it ends up needing it to reproduce. So these types of ecological mismatch that's driven by climate effects can be quite a cascade of effects throughout an ecosystem to lead to disruption and often species effects and potentially extinction. But this kind of understanding of how climate change and anthropogenic change is impacting organisms and communities has been facilitated by legacy data. And that legacy data takes a lot of different forms. One of the ones that I use quite a bit is museum specimens. And so I'm excited for tomorrow's physical object session. So come hang out. In my master's work in particular, I used herbarium specimens or pressed leaves from maple trees all throughout the East Coast to see whether we could document those phenology life cycle events over the past 200 years of collected leaves. And so that work wouldn't have been possible without someone going out in their backyard and picking up a leaf in the early 1800s. And so that type of work, we were able to show that as climate was warming, the timing of the color change in maples was shifting. Another piece is that these museum specimens aren't just documenting the place and time that an organism occurred, but all of these different features of what's going on at that point in time for that organism. So we get information about phenology. We also get information about genomics. We're able to extract DNA from these museum specimens to see at a molecular level or at a genetic level, how have these organisms changed over the past couple of decades or past 100 years or so. So recently, some work that I've been working on now has been looking at museum specimens of mouse to see how their genetics has changed as they've moved through different environments as invasive species. And using that data, we're actually able to pinpoint for the first time where mice actually come from, because they're so ubiquitous around the world as an invasive species that we actually didn't really know where they came from. Museum specimens play a big role in this. And so this work has really been influenced by this endowment of specimens that, we've inherited from the past. But there's a variety of other forms that this data takes as well. So one other key source is field notes, and growing more and more is community science data. So other work has relied on just sort of the casual note taking that people have done about, this is what's in my backyard today, and this is what it looks like, and this is what I've seen on my walks. And putting that data together can be quite challenging. It's not necessarily in a standardized data format, right? It's just in handwriting often. So we've done some work to think about how we can synthesize written notes of nature into usable data products that can be used to really investigate how changes to the visible environment we inhabit have occurred over the past time since we've been keeping track. And similarly, the community science space has really enabled people who previously hadn't necessarily been able to contribute directly their observations of how their natural world was changing to these data sets that are helping us see these changes. So recently we've treated iNaturalist images as documentation of phenology as well, and looking at insect communities and how their demographics might be changing. As you can really appreciate, this work relies on a suite of biodiversity data resources that really enable it. And it comes in a variety of different formats. You've got free text, you've got images, you've got tabular data sometimes. And so putting it together can be quite challenging, but really illustrative of how our environment is composed and what that looks like over time. But more hidden, perhaps, is the extent to which this work relies on open earth science resources as well. We can't necessarily say what is causing a change in an organism without being able to tie it back to some of these mechanistic relationships with the climate or with relationships to other biotic and abiotic variables. So being able to access these data really enables an understanding of the true drivers of these effects. Perhaps even more hidden, but less probably to the people in this room, is the infrastructure that goes into this. The data platforms, the digitization efforts, the science gateways that enable access and reuse of the data that is so critical to answering and asking these questions. And finally, collaboration. There's so many different people across a lot of different fields that enable this work, both at providing data, collecting data, synthesizing data. And for each of these works, there's been perhaps obviously collaborations at the academic level, but less clearly collaborations with government agencies and officials, particularly Smithsonian in this case, nonprofits. In particular, we've worked closely with Mohonk Preserve, which is a pretty small field station in New York that has a wealth of notes about nature, and the public. So much of our data is coming from your person next door who is observing all of these important things that are affecting their daily life, but could be synthesized to explain global trends. So another aspect of my work that's been growing in recent years has been providing tools to maintain a data legacy and provide a data impact for the future. So by making tools available to members of the public for sharing data that they collect and see, we're able to preserve this record for research now, but also enable future creative research usage of this data to understand the current, the present, and the future. And so this has looked like a lot of work with a tool called Anecdata, which provides data management capabilities for communities collecting data. And recently, we've been using so much of the projects that were collected or planned for individual community usage to be placed into federated data repositories for use beyond what the original scope of the project was. So we've been putting first our organismal data into things like GBIF. And so we now have more than 250,000 observations from these platforms. And each of these photos has a little bit of a story with it. And I don't have time to get to all of them. But up in the upper right for you guys is a little stingray. And it doesn't necessarily look like much, but this is a photo of an endangered stingray species for which there were no available public, freely usable photos of it in the wild. So all photos that existed were of dead specimens or ones that had been harvested for various reasons. So by providing natural photos of these organisms, we're able to understand what they're doing in nature. So synthesizing this data has given us a window into what our communities are interested in and how we can support using their data to support their own goals. So what's next? I didn't yet get to the genomics part, right? So more and more my work is using genomics tools as well. Like as I mentioned with the mouse specimen, we're able to take genomics information to predict how environmental change might affect species themselves at a molecular level. So every organism on Earth has a trajectory that could lead to persistence and survival, speciation or pretty major change, or extinction. And understanding what the genetic correlates of each of these states might be can help us understand how to have resilient ecosystems in the face of climate change and preserve key critical environmental effects and ecological effects on our ecosystems for healthy ecosystems for the future. So what? So I think there's kind of 3 take-home messages that I would like for you to take away from this. Creative uses of our data endowment can look like a lot of different things, and it can lead to critical insight for the planet. And many of us are data providers, and we hope for these kinds of creative uses of data that are ways that we could not even imagine, right? But enabling that through open tools is one of the ways that we can support research for the planet. And I think we really have a duty to the present and future public to collect and share data in ways that is open, but also prepared for reuse. So prepared in ways that can be used by communities to ask and answer critical questions to understand their environment and how it's changing. And finally, understanding climate change requires synthesis and collaboration, not just in fields that have traditionally worked together, but really across all of the disciplines. Almost always, I know we've got some astro people in here, we're all dealing with science that's affecting the Earth, which is a place that we all live. And so collaborating across fields is a way for us to really share our insights to better understand how these interconnected processes are being affected and could potentially be investigated in ways to understand our future and our present. I can't get to everything I want to talk about, so come see me at the poster session. I'm happy to answer any questions or talk further about any of these little pieces that I didn't quite get time to get to. So I'm really grateful again to everyone, the Raskin Scholarship and the ESIP community, and I'm happy to take any questions.
So that is a great teaser to go to the poster session so that you can ask any questions that you have. But we have to keep moving because we do have more sessions this afternoon. So it is now my pleasure to introduce this year's Charles Falkenberg Union Award recipient, Angelia Seiferth. The Falkenberg Award is jointly sponsored by ESIP and AGU. Dr. Seiferth is a professor in the Department of Plant and Soil Sciences, one of the founding directors of the Center for Food Systems and Sustainability, and the Associate Dean for Research in the College of Agriculture and Natural Resources at the University of Delaware. She earned her Bachelor of Science degree in Environmental Science and Chemistry at Towson University, her PhD in Soil and Water Sciences at the University of California, Riverside, and was a postdoctoral researcher in the Department of Environmental Earth System Sciences at Stanford prior to starting her faculty position. Angelia's research focuses on using tools and concepts rooted in soil biogeochemistry to develop sustainable ways to lower human exposure to harmful contaminants in food. Her research also includes understanding mineral control on below-ground carbon cycling in coastal environments that are prone to sea level rise and storm surge.
Thanks, Susan, so much for that introduction, and thanks to all of you for sticking around. I'm happy to be here. Let me figure this thing out. So first of all, before I start, I just wanted to express my sincere gratitude to ESIP and AGU for selecting me for the Falkenberg Award and to the Falkenberg family for establishing that award. I'm really, truly honored to be named. And so, as Susan mentioned, a lot of my work is focused mainly on looking at contaminants and ways that we can lower human health risk. And so today I'm just going to talk with you a bit about some of the work we've been doing where we're trying to visualize some of these chemical constituents in food and in soil. And I'm going to use rice as the example. So many of you probably know that rice is prone to arsenic uptake. You've probably heard about this before. There's been plenty of talks at meetings like AGU about it. And so the way that we grow rice in flooded soil is part of the reason why it's prone to arsenic uptake. It also is because it's a silicon accumulator, and the chemical form of silicon and the chemical form of arsenic in flooded soil are similar. So it sort of sneaks in along that pathway. And because of those two reasons, rice is really prone to arsenic uptake. In addition to just naturally abundant arsenic in soil, the process of moving arsenic from, say, contaminated groundwater aquifer and using that to irrigate rice can also lead to elevated arsenic in the rice. And so for me as a biogeochemist, the rice root rhizosphere is a really interesting place to look because there's these very steep gradients in oxygen that set up gradients in chemical constituents from what I call the bulk soil and the rhizosphere, that zone of soil right around the roots. And rice, like many other aquatic plants, they essentially leak oxygen into the rhizosphere. So as iron oxides reductively dissolve, they release the arsenic from the bulk soil. The plant is sucking solutes from the soil into its roots, and it provides a way for oxygen to react with those reduced constituents and oxidize them on and around the rice roots. So part of the visualization that we do is really looking at those roots. So if the iron oxides are precipitating on the roots, it's one way that we can sort of maybe prevent arsenic from getting into the plant. But if you look at the picture of the roots themselves, there are plenty of areas that are white, and those are just the roots themselves without oxygen or without iron oxides on them. And so arsenic can move freely into those portions of the root. And so from a food safety standpoint, we're concerned about the grain, but if we look at the mass balance of arsenic in the rice plant, most of it is associated with that iron oxide, what I call plaque. And so it's important to understand, is it a source of arsenic or is it a sink? So these are the sort of questions that we're trying to probe in my group. And one of the ways that we do that is through synchrotron X-ray fluorescence spectroscopy and imaging. And so what you're seeing here is a little depiction. We basically shine X-rays of a certain energy on a sample, and we can do that with a soil, we can do that with a rice plant tissue, and we get what I like to think of as a chemical photograph of where the elements are. And so what you're seeing on the top left is a soil thin section that we prepare from rice soil. On the right is sort of a zoomed in chemical photograph or X-ray fluorescence image where you can see where the root had been, and then a halo of iron oxide or iron around it. And then we can look at the arsenic that's associated with that iron. And as we move closer and closer to the root, we see more and more oxidized arsenic. As we are farther away, it's more reduced and it's more prone to being dislodged and possibly mobilized. So using that tool, we can probe different areas in the rice rhizosphere and look at things like arsenic and how what you see on the top left is a cross section of a root with a lateral root emerging. And so we think these lateral roots where they sort of push out of the vascular tissue are ways that solutes can freely get into the rice without having to get restricted by some of the plant processes. So if you look at the first X-ray fluorescence image, sort of in the middle of the slide there, the red is the arsenic. And so that is getting right into the vascular tissue. In addition to just looking at the specific elements, we can also look at how different forms of arsenic will change in space across the rhizosphere to root interface. And so that's important because for some of these constituents, some are more toxic to humans and some are less toxic to humans. And so by understanding some of these mechanisms of how the arsenic gets in, we can start to develop ways to reduce its impact on the plant and on humans. One of the ways that we have started to visualize how these roots develop and how this iron plaque is forming is using these vinyl films or sheets as passive sampling devices. So as plants grow, the roots are changing in space with time, right? So they're exploring more and more parts of the soil. And so if we want to understand how that happens, we can either dig up a plant and analyze it, but then we can't then sample that plant later, right? That's a destructive sampling. But using this technique, we can actually put these sheets in and pull them out and either take just a scanned optical image or again, use this X-ray fluorescence imaging to look at the spatial distribution of the iron and associated elements like arsenic. And this we sort of discovered by accident in the lower left, you see what we call an iris film. So this is a passive redox monitoring tool that we've used in rice paddies to try to help farmers decide when they should drain their fields to minimize arsenic risk. And so if the roots happen to grow on the face of this, on sort of the white areas of the root or of the film, we discovered that we can actually get really beautiful images of what the roots look like. And so both of these images here, this is just the vinyl sheet where it happened to capture roots growing along it. And we see those iron oxide deposits that we can then image. And again, this data suggests that these lateral roots are really hot spots of biogeochemical activity. Going a step further, we're also interested in how different soils respond and behave. And so we have here just an example where we have 5 different kinds of soil that vary in texture and some other chemical properties. And we've used this technique, and every week we can go in and we can have a film. We can pull it out and replace it and try to do the exact same spot and analyze it. And what you're seeing here as you go from the hot to the cool colors is each week of iron deposition on the roots. And so this is just an example of the kind of data that we get, very detailed spatial information and also in time. But as I mentioned, all of this is really to try to develop ways that we can mitigate or minimize human health risk associated with contaminants in rice. So we built this rice facility at the University of Delaware where we can do controlled experiments on the plot level. And we look at sort of the whole system. So if we add something to the soil to minimize arsenic, we're also going to affect the greenhouse gas emissions. We're going to affect the microbial community. So we look at all of those things and we work with farmers to try to develop strategies that they could actually utilize on their fields. And so just one example of this is, as I mentioned at the very beginning, the silicon and the arsenic can compete. So if we can take an underutilized silica-rich rice residue like rice husk and add it to the soil, it provides enough silicon to compete with the arsenic for uptake. And that's this data that you're seeing here. And I apologize, I realized that my legend is missing some letters here, but the bottom line is that we can either decrease the total amount of arsenic going into the grain, or we can affect the inorganic arsenic, which is the more toxic form. And then we can take those grains and actually look at where those different species are forming. And so now instead of the rhizosphere, these are actual rice grains that we've done thin sections on. And as you increase the silicon, you change the distribution of the arsenic within that grain. And so if you look at the far right here, we're essentially moving the arsenic from the bran and putting it into the endosperm. And what the reason that happens is because we're changing the speciation, and the different species or chemical form of arsenic is getting deposited in different places within the grain. So just one example of how we can use this, this chemical tool to sort of visualize where these constituents are ending up, and in our efforts to try to minimize human health exposure. So I'm happy to talk. I know that was a lot of technical information, but I'm happy to talk in more detail about any of these if you're interested. I just want to give a big thanks to my very hardworking research group. Without them, none of this is ever possible. Also, the synchrotron sources that we use and my collaborators there at the national labs, as well as my funding sources. And again, thank you to ESIP and AGU for the Falkenberg Award. Thank you all.
Huge thanks to both Angelia and to Alexis. It's really great to get their perspectives and to see where their work is headed and how data is so critical to what they're doing. And we're just really excited to have both of you here with us this week. So to keep us on schedule, we have 2 more rounds of awesome breakouts this afternoon. So I want people to move on back upstairs or to the amphitheater, wherever your room is. And don't forget to join us for the research showcase. It is in the same location as last night's reception, down at the end of the Emerald Corridor. And we have a record number of posters and looking forward to a great afternoon and evening. So see you over there.